Good afternoon, everyone, and welcome to MSK CC Science Spotlight, live from MSK in New York. My name is Andrea Sheeting, and I'm a faculty in the immunology program at Sloan Kettering Institute. This will be our last and closing seminar of Science Spotlight for the season. As you may know, these webinars were born out of necessity due to COVID, and over the last few months became a wonderful way to stay connected during the shutdown and to learn about each other's exciting research, because in the end, it is the love for science and the search for cures for our patients that unites us all here at MSKCC. There will be a Q&A session after this presentation, and if you have questions during the talk, please email them to the Office of Scientific Education and Training at oset@mskcc.org or by tweeting at MSK-education. And because it is today the last day of the Science Spotlight seminar series, it is my distinct pleasure to introduce the person who initiated the series, Dr. Joan Massagué, as our seminar speaker today. Dr. Massagué is the director of the Sloan Kettering Institute and a member of the Cancer Biology and Genetics program. Joan is an internationally recognized leader in the study of TGF-beta signaling and cancer metastasis, and has published many of the seminal papers in these areas. His talk today is entitled The Logic of Metastasis. So Joan, welcome, and thank you for concluding the seminar series. You take it from here.
Thank you very much, Andrea. It's a great pleasure to participate in this series and to talk about something that it's a key problem and challenge in cancer. Understanding the biology of cancer, and more importantly, treating the disease, metastasis. In spite of tremendous progress on many therapeutic fronts over the past several years, metastasis remains a formidable challenge. Today, we are going to take a look at a number of vignettes based on papers that were published earlier this year by the lab. And I will try to extract from them a common thread that will try to show how there is an operating logic to the metastatic process, how there is not so much hocus pocus and unexplained complexity as we thought before we knew a little bit. Metastasis manifested in people the way this slide describes. I like to use it. I've used it before. It is the end product of a selection process whereby cells that left the tumor very early, before the tumor was diagnosed, and managed to survive. The vast majority of cells that try this die, but a few survive in different organs, will over time find a way to emerge and regenerate the tumor at a distance. We know many things about metastasis. Some of the most fundamental ones are that dissemination can occur very early. Yet overt metastasis frequently occurs late. Metastasis-initiating cells, the cells that have the ability to do this, very frequently enter dormancy that can last just a few months, or sometimes years, even decades, and then somehow grow to overtake the host organ. Genomic heterogeneity is a source of metastatic traits, but phenotypic plasticity, we've come to learn, is the name of the game. And we are going to explore this during my talk. These two sources of heterogeneity, of variegation, under the pressure, the selective pressure of a microenvironment that constitutes the various bottlenecks of the metastasis cascade, give rise to metastatic populations that are there, challenging us to treat them and eliminate them. What is that we would like to know? Well, a lot, but just to focus on what we are trying to achieve progress on today, we would like to know what is the origin and nature of these metastasis-initiating cells. Are they particular entities, or they are just part of the heterogeneous morass of the primary tumor repopulating distant sites? How do mix enter dormancy? Is that a passive event that they endure because they are not welcome in distant organs? How do they evade immune surveillance now that they are not protected by a tumor mass? And eventually, how do they develop outbreaks? And of course, very importantly, at the end of all this, are there new druggable targets that could be used to treat cryptic metastasis, metastasis before it is full-blown? When metastasis is full-blown, we have and applied to it all the major forms of therapy, surgery and radiation, and very especially targeted therapy, chemotherapy, immune therapy, immune checkpoint inhibition therapy. All of that is being applied to metastatic disease most of the time. Of course, also during the adjuvant treatment in the adjuvant setting. In the adjuvant setting, treatments in order to try and eliminate the disseminated cells, the invisible metastases, the cryptic metastases, we are treating that with the same drugs that were developed to treat growing tumors, primary or metastatic overt tumors. And yet we do that without knowing almost anything about the biology of these states of dormancy and cryptometastases, which is what I would like to explore with you. Now, phenotypic context determines cellular responses of all kinds, something that we learned early on in my lab when we were studying the action of TGF-beta and the infamous role of TGF-beta as a dual agent in cancer. TGF-beta is a powerful tumor suppressor for the initiation of and progression, early progression of tumors. However, tumors and tumor cells that manage to avert these suppressive actions, cytostasis, apoptosis especially, will be able and find a way to use TGF-beta as a pro-metastatic signal, acquiring activities that they were never seen before in normal cells responding to TGF-beta. Their phenotype has imparted on them the ability to manifest responses and behaviors that are unique to that phenotypic state. Some details and highlights. When we were studying how TGF-beta imposes cell cycle arrest in epithelial progenitors, a very important response in development, stem cell and progenitor cell biology, we were able to define a tidy pathway from hormone through receptor and mediators to target genes whose products encode the actual direct biochemical inhibitors of the cell cycle machinery. That was fine, but it did not apply to what one would have thought to be a germane question: how is TGF-beta inducing apoptosis? TGF-beta induces apoptosis when cells, progenitors, have acquired oncogenic mutations, typically in RAS or hyperactivation of RAS by any other mean, a constant in solid tumors for sure. There we went on to find, after many years of puzzling over this, that the reason why these cells engage in apoptosis and are eliminated in response to TGF-beta has to do with the fact that TGF-beta, in the presence of a strong MAP kinase activity, induces expression of a SNAIL, a gene that, mediates, transcription factor that mediates the epithelial to mesenchymal transition, another phenomenon of pronounced cell phenotypic plasticity. But in this context, in the context of a progenitor that is meant to be an epithelial cells, that creates a clash of pathways and precipitates apoptosis. Yet if these cells progress, it's because they were able to eliminate these barriers, either by accumulating mutations that disrupt the TGF-beta receptors or the SMAD signal transducers, or by somehow decoupling the pathway from apoptosis. If they achieve that, and we now know how they achieve that, how they do the decoupling, now they get to use the pathway in their phenotype to accumulate responses that are supportive of their metastatic progression. As we first learned many years ago when collaborating with Elisa Geis to discover that TGF-beta is a strong promoter of osteolytic metastases by breast cancer. It induces expression in breast carcinoma cells in the bone of factors that hyperactivate osteoclasts to dissolve bone matrix to release more TGF-beta, thus establishing a vicious cycle of prometastatic osteolytic process. So, given these complexities, given that phenotypes are varied and respond differently, And that will play out in the context of a metastatic process. How are we going to understand this problem? The approach that the lab took was to use the mouse as a cell sorter, to screen for cells derived from human tumors or mouse tumors that would be asked to perform metastatic acts. For example, metastases, aggressive metastases to specific sites. We did that with cells isolated and derived cell lines from patients with advanced disease with multi-site metastases from breast cancer, lung cancer, and others. Or if we wanted to know more about the early stages of metastases and the cells that are able to seed those, then we would, as we did, do this experiment, select for cells derived from early stage tumors that have not yet learned how to metastasize effectively. For the first of these two examples, we developed protocols to isolate these cells, verify that they have strong proficiency to overtake lung, brain, bones, liver, and then through omics comparisons, identify the expression of genes, microRNAs, and anything else that anybody would be interested in that would be associated with and eventually shown to mediate the metastatic phenotype, moreover, whose expression in human primary tumors was associated with an outcome that completely matched what the phenotype, metastatic phenotype of the cells had been, and the genes driving it had been in the assay systems. This was successful, was productive. It gave us an entire list of genes that are associated with and mediators of specific metastatic steps. Extravasation, passage of cells from the circulation into diverse tissues, including the brain through the blood-brain barrier, seeding, resisting the rigors of having entered for the first time these cells, a hostile environment, and being able to set states there for their eventual growth, stem cell niches and whatnot. And finally, in the phase of overcolonization, genes that these cells use to engage the surrounding tissue, the components of the microenvironment, productively to co-opt them, enslave them, to favor, support the now aggressive growth of metastatic lesions. This was productive not only at this level, but also at other levels, providing answers to the question, how does TGF-beta induce metastasis when it does? Target genes of TGF-beta were found among those that had emerged independently as mediators of metastasis. And in a number of instances, genes that became targets of ongoing and recently completed and under manuscript preparation clinical trials. One of my favorite examples is this one from brain metastasis, work initiated by Paula Bosch, Don Nguyen, and Manuel Valiente, and more recently in this paper, not so recent anymore, by Qing Chen and Adrien Bois, that pursued the question of how do cancer cells that extravasate from the purple capillaries on the left as green labeled cells here, how do they deal with that environment? Well, first they are detected by highly reactive astrocytes that are out there to kill them, to kill the intruders by production of killer cytokines. And the metastatic cells were found to produce SERPIN inhibitors of a protease that enables the cleavage and activation of the fast ligand killer cytokine. But when the cancer cell had done that and been able to survive there, then it did not hesitate to, if it had the tools to do it, to make a deal with astrocytes, to connect and form gap junctions through connexin 43 and protocadherin 7 to obtain survival and outgrowth benefit. And we went on to learn that the one thing that passes through, flows through those gap junctions from the cancer cell to the astrocyte was CCAMP, the mediator dinucleotide of the STING pathway that is activated in cancer cells on account of their genomic disarrays and double-stranded DNA floating around in the cytosol or being in fragments in the cytosol. That activates the initiation of the pathway. However, cancer cells have selected against an active pathway that would expose them to immunity. They are happy, however, to pass CGAM to astrocytes that will complete it for them and delivering salutary levels of interferon alpha and TNF for this action in the cell. Based on these insights, the physician scientist says, hey, I have an idea. How about trying to develop clinical trials around known inhibitors, negative regulators of gap junctions? And so this is what had been and has remained to some extent the thrust of metastasis research in the lab. It's a very gene-centric thrust. We are not asking, and what cells are these? But rather what genes give some cells these abilities, and could we do something about them in the clinic after we learn about them in the mouse models for their biology and biochemistry. But eventually it was the poignant reality that when metastasis is at that level, it's better to have prevented it from reaching the overt metastatic state that led us to consider whether we could achieve similar progress by interrogating cells that have not yet graduated to the overt metastatic state, but have the ability to be metastasis initiators. We know a few things about these cells. I already mentioned that before. They have tumor regenerating ability. They may well go on to regenerate the tumor after months or years. They can enter quiescence. They are found and described by pathologists as being in tissues in a state of quiescence. Evidently, that will be reversible because they will grow again. They are immunoevasive, even though they are exposed to the immune surveillance to the four winds. They are not protected by their tumor of origin anymore. They are somehow left alone by immune surveillance. And they are chemo-resistant, at least to the therapies that are being applied in the adjuvant setting in an attempt to cleanse the body of these intruders. It was Srinivas Maladi, now at UT Southwestern, as a faculty member there, who took on this task, and using cells derived from early stage 1, 2A, lung adenocarcinomas and breast carcinomas, he went on and selected cells that had the capacities that we were looking for and we were screening for. Cells that can install themselves in the tissues of a mouse, any tissue, brain, lung, adrenal, kidney, and live there without very frequently mounting a full-grown metastatic colony. In most of the mice, the cells would just sit there, not one would proceed. But if you recover these cells and put them back in culture, they would grow very well, and they would do it again if reinjected in a mouse. In other words, these were not senescent cells that were dead ends. These were cells that have reversible growth potential and the ability to survive in mouse tissues. Srinivas went on and interrogated these cells phenotypically and molecularly to make a series of fascinating observations. How these cells, indeed, they live singly, although on occasion there are small clusters. If they have formed clusters, the clusters accumulate Ki67. They were proliferating the day that you examine them. But the single cells are not proliferating, are truly quiescent. Molecular analysis by RNA-seq, and here Western blot of the representative genes, illustrated that the cells present transcriptomes typical of progenitor cells, stem progenitor cells of the tissues in which the tumor originated, breast and pulmonary epithelium in this case. SOX2 and SOX9 being prominent among these genes. And in the case of the lung, for example, SOX2 is a well-known and important transcriptional regulator of the early stages of lung budding, lung formation, tracheal branch, bronchiolar branch, and eventual branching of the growing lung. And SOX9 is expressed in later, more distal elements during development, and also, as we shall see, during regeneration. He also noticed that another property that these cells had was to downregulate, when they were quiescent, a number of determinants of sensing, being sensed by the immune system, specifically ligands for the natural killer cell receptor, NKG2D. the ligands for these expressing the cancer cells were very downregulated when they entered quiescence. That gave the idea or the hypothesis that perhaps the cells are quiescent because that's what they do, that's what they have to do to survive the constant vigilance of NK cells. The experiment that is shown here was done 2 arms after in mice that had been inoculated and had harbored dormant cells for a full month. 2 arms, one just untreated and the other depleting NK cells from these mice with GM1 antibodies or anti-NK antibodies, to find indeed that whereas in the untreated arm, there would be occasional, in some mice, an occasional metastatic tumor growing here and there, as in people. And most mice would have not a single tumor. The mice depleted of NK cells would present with massive tumoral growth in multiple sites and multiple lesions. So that and other insights led to these views, these news about the properties of metastasis-initiating cells. They are stem progenitor-like of some kind. They enter quiescence not as a passive reaction to an environment that has lots of TGF-beta, as others had already indicated that TGF-beta might be indeed a mediator of quiescence here, but rather very proactively. They express DKK1, an inhibitor of Wnt. They're favoring mitogen. Ambient Wnt levels, therefore, are not acting on these cells, and these cells, therefore, fall into quiescence. They also are hypersensitive to inhibition, growth inhibition by TGF-beta. And in entering quiescence, they become invisible or much less visible to immune surveillance, at least for NK cells, and we believe also for CTLs. What else? How do progenies evolve after these cells do manage to mount an outbreak? For that, we turn to the powerful technology of the day, single-cell analytics. These led by another postdoctoral fellow in the lab, Ashley Loughney, now a faculty member across the street at Weill Cornell. In a fantastic collaboration with Dana Pear and Charlie Rudin, we took on one of these 2 model tumor types that we have been studying, lung adenocarcinoma, and analyze the content, the cellular content, based on single-cell analytics of tumors versus normal lung epithelium versus metastases to various sites. A number of things were learned immediately. That is that the carcinoma population is minority in these tumors, only about 20%. It varies from tumor to tumor, but always minority. The most abundant population in these tumors are actually immune and inflammatory cells, as you can see. And there is also, of course, presence of niche cells, epithelial cells, cancer-associated fibroblasts, and other mesenchymal components. We started to ask questions about the relationship that might exist between this population, the epithelial population, now focusing just on the epithelial component, the carcinoma component, their relationship with the normal cellularity of the lung epithelium. And the finding was immediately clear and striking. How this population of cancer cells contain very readily distinguishably the phenotypes that define not only the cells of the homeostatic lung epithelium, alveolar epithelial cells type 1, type 2, basal cell, clump cells, and ciliated cells, but also cells that are found when the lung epithelium is damaged. They emerge as progenitors that will be in charge of reconstituting and regenerating that lung epithelium following that damage. So it gave us the impression that tumor cells are commandeering normally silent regenerative programs, possibly to drive malignancy. That was in primary tumors. Among the epithelial cells in the metastases, what we found was an even cleaner version of these. I say cleaner because in the primary tumors, all the gray cells in this force-directed layout of normal and primary epithelial cells, many of these gray cells have a mixed match of phenotypes. Only the ones in color present pure, if you will, or at least very crisp phenotypes corresponding to the known normal cell types. In metastases, there is higher purity and overall higher earlier stage abundance of earlier stage phenotypes than in primary tumors, as if what has been selected here had a high level of fitness to adopt all the phenotypes of the developmental and regenerative continuum of the lung epithelium. SOX2 and SOX9, you can see them here. SOX2, a representative of the foregut and developmental stages, as well as the proximal components of the pulmonary epithelium, is abundant there, whereas progressively as the epithelial fates correspond to more distal ones, we find SOX9 dominating. Among the most primitive double negative for SOX2 and FOX9, we find SOX17 cells that is yet another very early stage endoderm transcription factor, and included here also cells that are positive expressing vimentin and SNAIL1, markers of EMT, and these cells are very lowly proliferative. So even in growing metastases, we have cells that are reminiscent of the quiescent state of dormancy, along with cells that are proliferative and along a seeming path of development. Moreover, we also notice that as cells proceed through these phenotypes, the determinants of sensitivity to immune recognition vary tremendously. The quiescent state has features of that would predict resistance to NK cells, also have a high level of class 1 HLA protein expression, low levels of ULBP, RET, ligands for the NK cell receptor, ligands that appear when these cells are in an early stage, are early stage progenitors in a proliferative state. In this state, SOX2 high, SOX9 low, it's a state that you would predict is highly vulnerable to NK cells, whereas later stages, the SOX9 rich, might be less vulnerable. Indeed, what we find that accumulates most readily in metastases are the SOX9 cells. This is all portraits of the cellularity that exists in these tumors. There is no time sequence to them. However, remember the model that I presented a moment ago and the experiment of allowing these cells, these latent lung adenocarcinoma latent metastatic cells, to enter latency or to exit from latency spontaneously, or to do it massively if one depletes the mice of NK cells. So Isley also analyzed cells extracted from each one of these spontaneous stages: the dormant, the incipient metastasis, very micrometastatic, the lowest level of increase in luciferase expression by these cells in situ, or cells that have escaped and succeeded at mounting a strong metastasis. The force-directed layout of the phenograph constituents of these populations is shown here in the middle, showing the route or the trajectory of these various fates. And to the right, you see the expression superimposed on these maps of SOX2 and SOX9. How SOX2 indeed characterizes the cells in the dormant as well very early stage of emergence from dormancy, whereas the scapes, the macrometastatic states, are dominated by SOX9 expressing cells. Suspecting, based on all these, that there is a differential pruning, selective killing, elimination of these cells as a function of their immune susceptibilities by NK cells at least, we went on to the experiments that show that that indeed was the case. Spontaneous outbreaks are rich, as I said a moment ago, for SOX9 positive cells. But if the outbreaks have been enabled by us depleting NK cells from these mice, the outbreaks are very rich in SOX2 positive cells. Experiments of Q incubation in vitro of activated NK cells demonstrated on the right that indeed SOX9 cells become more abundant, not because they are not killed, they are killed, but much less so than the SOX2 cells that are decimated by the action of NK cells. So that leads then to a growing compendium of insights that begin to give us a sense that metastasis is a dynamic interplay between developmental plasticity and the immune-mediated elimination processes. And the two together shape the cellularity and the composition of phenotypes of the resulting tumors. What else? What functions do metastasis-initiating cells require to initiate an outbreak? As the balance of mitogens got them back into the cell cycle, do they need something special there other than somehow evading immunosurveillance, getting lucky and not being picked by an NK cell? Well, the answer to this question is yes, they need something else. And what that was came from just watching these cells under the scope. Images developed by Manuel Valiente some time ago were striking, the kind that you see them once and you never forget. When these cells, green labeled metastatic cells, are inoculated into the bloodstream of a mouse, human cells will extravasate in the organs for which they are meant to be highly metastatic, in this case, the brain. And when they do that, they don't walk away, they stay attached and hugging, as you can see, the vessel very tightly. And if they grow, they will do that by remaining attached to the vessel in many types of models, most of the models, with some interesting exceptions that we have developed and studied. Here we are looking at the result of 3 cell divisions by one of those cells, spreading on the capillaries, 8 cells, and we went on to identify an adhesion molecule that allows these cells the ability to spread in this manner on the capillaries, L1-CAM. L1-CAM, a member of the cell adhesion molecule immunoglobulin and fibronectin repeat family, includes many others, but L1-CAM was remarkable. There was an extensive literature about it describing how this molecule normally found, or normally thought to be found only in the brain, is frequently found in solid tumors of every kind, carcinomas, melanomas, sarcomas, and the level of expression or the proportion of L1-CAM positive cells in these tumors over and over had been associated in numerous papers with outcome. Being a cell adhesion molecule, it was assumed that it would have to do something with migration or metastasis, but nobody, of course, knew what. Now we find it as the key component expressed by metastasis-initiating cells, and they use it to spread in this dramatic fashion on capillaries. If we knock it out, in cells that are going to be entering the bloodstream, these cells are still navigate through the bloodstream, extravasate, still stick around vessels, but they cannot spread. And with that, they fail massively to form metastatic colonies. This is seen not only in lung cancer models in the metastatic to the brain, but systematically we've been seeing these breast, colorectal, renal carcinomas, metastatic to any of the sites that they are metastatic to: brain, lungs, bone, and liver. Why? What do these cells get out of attaching themselves in this manner together with L1-CAM, a known collaborator of integrins, but again, collaborating to do what? The answer to this came from studies of Embraer with contributions by Yasmin Kagusus, who is a grad student in the lab. Embra is now a faculty member at the University of Illinois. Embra went on to show that when these cells attach themselves on capillaries, they activate in conjunction with integrins and via anchoring to and the PALK integrin-like link kinase set of events, chain of events, or pathway, the activation of YAP and MRTF, mechanotransduction activated transcription factors that are strong promoters of cell proliferation in progenitor cells. Even though these cells are cancer cells, even though they are bicapillaris exposed to oxygen and nutrients, they need the green light of YAP transcriptional activation in order to have the ability to engage in productive growth. And the images above are of real human-derived metastasis tissue samples, verifying there what Emre was also able to show in the mouse models. So now we've learned, in addition to the stemness, self-imposed quiescence, and immunoevasive nature of these cells, that these cells are capable of outgrowths that undergo selective immune-mediated pruning and course proceed along the developmental path and regenerative cell phenotype path of regenerative processes. How outbreaks have special requirements. One that we have found, we're sure it's not the only one, but one that seems to be important and fairly universal, not unique to any given tumor type or organ site, is L1-CAM as an agent for activation of YAP1. And what else? Well, at this point, a question that came up that was provocative is these cells, which are stem-like and have the ability to generate tumors, might there just be cancer stem cells, the name given to adult stem cells that acquire mutations and go on to initiate tumors? A classical example among solid tumor biology is colorectal tumors. The LGR5 positive cells at the bottom of the crypt, stem cells of daily homeostasis, of reproducing, replenishing the epithelium through natural turnover. These cells are the ones that, if they receive mutations that hyperactivate the Wnt pathway, will go on to form an adenoma and then graduate to carcinoma through the accumulation of additional mutations. They are cancer stem cells, tumor-initiating cells. Are mixed metastasis-initiating cells, simply cancer stem cells that acquire a few additional prometastatic traits. For that, enter Karuna Ganesh, and you've heard the story, some of you, their colleagues, now as a faculty member here at SKI. Karuna reproduced the results of many before, which is the observation that sure, L1-CAM cells are present in primary tumors, are absent in normal tissue. Moreover, they are enriched. They are not very abundant, but to the extent that they are present, they are enriched at the invasion front, including, if one looks closely, at these clusters of cells that are attempting already to enter blood vessels for presumably metastatic dissemination. There is a further enrichment of the cells in liver metastasis, and we are looking here at patient-derived tissues just stained for L1-CAM, and further enrichment if one subjects these patients to neoadjuvant therapy. Moreover, Karuna went to interrogate these cells for the ability to perform deeds that are considered the functions and the abilities of stem cells, homeostatic stem cells, regenerative stem cells, perhaps. She separated from tissues that are from human lesions, L1-CAM high versus L1-CAM low by flow, and assayed for organoid formation. You know, the three-dimensional structures that remarkably recapitulate the structure if they come from normal, non-tumor epithelial cells, the structure of the homeostatic intestinal epithelium. And if they come from a tumor, they will reconstitute the cellularity of that tumor. As you see, it was the L1-CAM high cells, the ones that have higher ability to form organoids and higher ability to transplant tumors to mice. The L1-CAM negative cells were poor, and to the extent that they grew organoids, when Karuna analyzed these organoids, she found that these cells, through undoubtedly plasticity, phenotypic plasticity, had regained the ability to express L1-CAM. She knocked out L1-CAM, no organoids, no transplantation of the tumors. So marker and mediator of tumor regenerative capacity. He's looking at the organoids sustained for L1-CAM. The cells that initiate the organoids are L1-CAM positive. As the organoids grow, they begin to relegate the L1-CAM positive cells to the corners. And in the case of organoids derived from normal epithelium, to corners that look like creeps. Another remarkable astute observation of Karuna was when one dissociates these organoids, there is a massive burst of even higher expression of L1-CAM. These are organoids derived from normal human epithelium, intestinal epithelium, or from human colorectal tumors. He went on to decipher, to delineate how L1-CAM in these cells that can express it is kept silent by the action of the REST complex, the REST transcriptional repressor complex that is sitting on the promoter until these cells are dissociated or they lose adherent junctions by any other means, like, for example, knocking down EC adherent. Then the expression of L1-CAM is on, and these cells are seeking substratum to reinitiate their growth, as this scheme represents. What is the relationship to cancer stem cells, to LGR5 positive cells? Well, this tumor organoid initiating cell, work of others, Prisca liberali, is L1-CAM negative, even if it comes from is LGR5 negative, even if it comes from an LGR5 positive cells. The first thing that those cells do is to downregulate LGR5. So an organoid starts with an LGR5 negative, L1-CAM positive cell. It will reacquire LGR5 expression as a requirement for further development of the progeny. But the start is dominated by L1-CAM, and indeed, based on single-cell transcriptomic analysis of the cells and a flow sort map of sorts based on transcript levels, Karuna was able to show that the majority of L1-CAM positive cells are low or null for LGR5 expression. Because this was more and more reminiscent of regenerative biology, Karuna decided to look in epithelium to find, normal epithelium, to find that indeed there was no expression of L1-CAM unless you hurt this epithelium by providing, administering dextran sodium sulfate, an inducer of strong colytic response. One sees that there is massive appearance, mobilization within a few days of cells that have expressed L1-CAM and will remain L1-CAM positive for as long as the epithelium is trying to regenerate, to cover the gap, to give rise then later to the regeneration of the homeostatic epithelium with an entire tree of differentiated cells. These mice don't need L1-CAM normally. You can knock it out in the intestine and they will be fine. However, if you knock it out in the intestine and you hurt the intestine, these mice cannot seal, as done in this violin-Cree-driven knockout of L1-CAM using a conditional allele. The mice will not be able to regenerate and will die of incurable ulceration of the intestine. So these and other insights led to the vision that we are truly dealing with regenerative biology co-opted by cancer cells. How in normal epithelium, less in the case of colorectal cancer, now we are deeply investigating these matters in lung adenocarcinoma, and there are very interesting similarities, but also differences emerging. But in the case of colorectal at least, the emergence of L1-CAM is a result of a call to regenerate, to cover a gap in the barrier tissue. And that will occur again, not at the initiation of a tumor in the adenoma stage, where the tumor, the integrity of the epithelium is still fine at the cellular level, but as the growing lesion, now turned into a carcinoma, will begin to lose structure, epithelial structure, and that will be sent by malignant progenitors to express L1-CAM and to enter the circulation with that capacity that they will reuse happily to reinitiate, regenerate growth of the wrong tumor, of the wrong tissue in the wrong place, what we call metastasis. Now, is this all that the cells do as they extravasate? No, they do something else, and it's what you are seeing. These cells, right after extravasation and during extravasation, they have had to resort to another dimension of phenotypic plasticity, that of EMT. When the cells extravasate, they are in EMT and they are migrating up and down the vessels. Here is using a trick that Sitting Gan has adopted in the lab, labeling, loading, or allowing the test cells, metastatic cells that will be injected into the bloodstream of a mouse, loading them with a diffusible, membrane-permeable version of M-cherry. And these cells now, once they enter tissue, they will be secreting that M-cherry and labeling all stromal cells that they enter in contact. As you can see on the left, these bifurcated endothelium, or rather capillary in the brain, has on the right been labeled by these metastasis-initiating cells that extravasated and walked up and down to the left branch, to the right branch, before it will find presumably a spot that it can call home, a stem niche composition upon which it will extend. It will use L1-CAM to proliferate and grow. So that was, yeah, we call this the trail of footsteps left by the cancer cell right before the tissue was extracted to be imaged by Sitting. Now, TGF-beta, our TGF-beta, is the most frequently invoked mediator or inducer of EMT in many contexts. Cancer, normal wound healing, gastrulation in the embryo, the nodal, TGF-beta nodal, driving EMTs. So for a long time, this had been known, but nobody had explained how an EMT is triggered by TGF-beta. There was enough going for it that made us react and want to understand how TGF-beta induces an EMT, because that is going to be very important in understanding the biology of metastasis-initiating cells. Jessie Su, when she was a postdoc in the lab until last year, noticed that along with SNAIL, the EMT transcription factor, there was another subset of TGF-beta target genes, encoding most of them fibrogenic factors, fibrogenic signals that will act on surrounding mesenchymal cells to produce large amounts of extracellular matrix, a wound healing response. This set of genes, 13 genes, osteogenic genes plus the SNAIL and Has2 EMT genes, are genes that TGF-beta induces best, most strongly, if the cells harbor a hyperactive MAP kinase pathway because of the presence of RAS inhibitors. I mean, activated RAS oncogene. She went on to conduct with Chuck David, another postdoc in the lab at the time, a shRNA screen trying to identify factors, transcription factors, that would somehow cooperate with SMADs to mount these very strong but RAS-dependent gene expression events leading to EMT. She found two, SOX4 was expected and not directly related to the mediation of the response, but the other was. The other is RAS response element binding protein 1, a almost forgotten effector of the RAS pathway. Very few papers on it. The RAS pathway has well-known transcriptional effectors, but REP was known to only a handful of labs and papers. It has this structure. It had been implicated in the limited number of papers in the integrity and migration of epithelia from Drosophila to cultured cell lines. And here it was, a factor that Jessie went on to show is essential for SMADs to activate this subset of genes. Not needed for other genes, represented here by SMAD7. They are just activated by SMAD, but SNAIL and the fibrogenic gene responses require REP1. And why that is, it's under investigation in the lab at the moment by Junho Lee and Liang-Ji Lee. But the effect and role of REP couldn't be more dramatic. You knock it out and you don't get an EMT, even though the cells remain responsive in many other ways to TGF-beta. As I mentioned, it's not only SNAIL1 transcription factor for EMT, but also a number of fibrogenic factors that are under the joint command of SMATs collaborating with REP1, TGF-beta pathway collaborating with RAS, orchestrating together a fibrogenic EMT, an EMT that belongs to the world of wound healing, an EMT where progenitors, epithelial progenitors will undergo it while communicating to mesenchymal components of the stroma, hey, you've got to make matrix for us, we've got to fill a hole here. This, in the context of tumors, leads to, as you can see in the image, abundant intratumoral fibrosis. And we extended this in collaboration with, wonderful collaboration with Kakhac and Tonakis. The same principle applies to the primordial EMT, that of gastrulation. Again, that is driven by nodal in cooperation with, through SMATs, in cooperation with REP1 to generate the phenotypic EMT, couple here, not to fibrogenesis, but to the genes that drive what the cells undergoing EMT are supposed to do, differentiate into mesoderm and endoderm fates. Going back to tumors, we see a tremendous impact of REP1 and what depends on it on the ability of cancer cells implanted in mice to form tumors or to colonize the lungs. With the knockout of REP, there is a precipitous failure to form metastases. We don't think that this is just because cells don't migrate. We think that the lack of fibrogenic response, of intratumoral fibrosis, is going to have a lot to do with the ability of these progenitors to grow and expand, not just to migrate. And these two is under investigation. Here's images of how tumors, even when size matched, the knockout, REP knockout tumors are devoid of intratumoral fibrosis, whereas the tiny, still tiny counterparts of normal, wild-type human lung adenocarcinoma cells growing as lung metastases in mice are already peppered with plenty of fibrotic tissue markers. So this is what we are learning so far, how metastasis-initiating cells are distinct from tumor-initiating cells, are endowed with high phenotypic plasticity. They can enter proactively quiescence. They, in doing so, acquire the benefit of immunoevasive dormancy, that they will come out of this dormancy and they will come out with a number of needs. Of course, avoiding immunity in this very delicate moment, foolish moment, if you will, in their existence, they need to resort to certain molecules, mediators of activation. L1-CAM activating YAP is the one that we find. The outgrowths undergo selective immune-mediated pruning, attracting immunity as they follow a developmental regenerative continuum that defines the cellularity of what results. Ideas about therapeutic approaches? Well, plenty out of this biology being dug out from obscurity. We have, of course, the classic approaches to therapy, target bulk cancer cells in metastases with targeted therapy, chemotherapy, radiation. Also, of course, now a sudden classic, target them with immune checkpoint inhibitors. Target has been attempted at the level of the stroma with inhibitors of osteoclasts, astrocytes, and others that support the unbridled growth of the metastatic tumor. However, the new insights give ideas about what we could do to attack the cryptometastases, the population of residual disease that will develop into metastases or was a metastasis, has been eliminated, debulked by the approaches on the right, and now has returned to the life of progenitors that will relaunch the metastatic growth. And time will tell which of these will be successes. I certainly hope that they will be. These are the colleagues that I have named as we went along. the current members of the lab listed here, the recent members of the lab that did a lot of the work presented today, with the current ones and collaborators without whom none of this would have been possible. And I thank you all for your attention.
Joan, terrific presentation as always, fascinating data and lots of questions. So we get started right away. A more general question to begin with. Is it known whether the cancer cells that leave the primary tumor site during tumor progression and the ones that might leave the primary tumor site later are similar? Do they have similar cell intrinsic metastasis-initiating capabilities? Are they similar or different? You would imagine that maybe the later ones are have an advantage in driving the metastasis, no? Is there anything?
That could be, but counterintuitively, work of others has led them to claim that the earlier ones are the worst ones. That is counterintuitive. I do not know how that would work, but you know, if the evidence is there, there is one more thing to explain. The cellularity, the composition of an early tumor may be more pure, if you will, less replete with cells that have adopted these mix and match phenotypes that may be less conducive for metastasis. As we see in the metastatic trees, developmental trajectories, those lesions, you know, it's a fairly tight ship with regards to phenotypes. And maybe that thinness is proportionally higher in cells from a very early tumor. This is speculative 100% in answer to this question. But I think that while the straight answer is it is not known, I think that there is room for the answer being, yeah, there are differences that are, you know, meaningful physiologically and perhaps even in terms of treatment.
And what's the contribution, genetic versus epigenetic regulation of driving that?
Many efforts have gone into identifying metastasis-inducing mutations, and that has been a dry well so far. From, of course, the high level genomic, massive genomic studies, TCGA and others, there have been mutations associated with metastases that were not present in the primary tumor. But at least the most prominent examples of those mutations have to do either with p53 mutations that generate more chromosomal instability, or mutations that are in metastases, most likely because they reflect resistance to therapy applied to metastatic disease. There are no mutations that I know of that says, aha, a gene that allows these cells to extravasate or to form a niche or to do business with the astrocytes is mutated and hyperactive. There should be out there, but it just hasn't come out. Rather is epigenetic, epigenetics. Now, epigenetics means what? I think that if I would make a prediction about what mutations might be related to metastases, they could be of two kinds. One, mutations that help these cells evade immune surveillance at the distant site, or B, mutations that may lock or favor the adoption of some of the phenotypes in this continuum that are more robust at retaining the metastasis-initiating capacity. And I remind us that many of the newer generation of cancer-associated mutations affect so-called epigenetic regulators, chromatin and DNA methylation regulators. It may well be that their function is not just to keep cells from differentiating, but to favor some of these complex series of regenerative phenotypes that are best at re-initiating the response, the tumor growth.
Another question in terms of the evasion of the immune recognition by NK cells. Is there a correlate in normal physiology, regenerative processes, or development for the elimination of cells by NK cells? This seems like an interesting handicap for the tumors that takes them a long time to escape.
I do. It's an excellent question, which is the answer that one gives when one doesn't have one. I find it very intriguing, but I do not know. An answer, conclusive answer to this question, if I understood it correctly.
Okay. So then what are the signals that allow a dormant cell to reactivate, especially in the spontaneous reactivation system? You showed the NK cell depletion, but there's a question about the spontaneous. What's the mechanism in there?
Let me preface the answer by the following, pointing out the following. For a long time, it had been thought, and many still think or assume, that dormancy is a permanent state where the cells go into dormancy and they spend 10 years like that. I think that's wrong. At least in the models that we've looked, it is not so. The moment you remove NK cells, that population will produce growth. And based on other lines of evidence, we know that the population is, these cells are coming in and out of quiescence. Probably it could be just as a result of fluctuations in local Wnt mitogen levels, TGF-beta levels, their level of expression of Wnt inhibitor. It's a metastable, if you will, quiescence. They will come out of quiescence. If they come out of quiescence, the NK cells will come and wipe them out. Mm-hmm. So this is why when you look, the only thing that, or the most of what you see is quiescence, but you always see those little clusters that never grow past 3 or 4 cell divisions, presumably because they tried and they were eliminated until one gets lucky, and then that is an outbreak. But there was nothing that on a given day, a Tuesday morning that was raining, woke them up mysteriously. Now, this said, we are very interested in knowing what I refer to as fluctuations in local mitogens and so forth, what that really is. And why we expect to dig out lots of fascinating biology about the niches that harbor quiescent cells, et cetera, all happening, by the way, in the perivascular space, right next to capillaries.
Okay. So then another question came in. Where are the traits for the organ-specific metastasis established? Already at the primary site or at the distant organ site? Because many cells leave the primary tumor, go to the distant site, but a lot of cells die. So yeah. Where's the survival count?
Both. We've seen examples and published examples where the microenvironment of the primary tumor, if it's very rich in, say, mesenchymal cells, particularly rich in that tumor. We saw that in breast cancer and published it. Sean Zhang, who is now a professor at Baylor. If the stroma is particularly rich and active with activated mesenchymal cells, fibroblasts, lots of CXCL12, lots of IGF, that selects in that population those cells that are better at thriving with these cytokines. When these cells abandon the breast tumor and circulate and arrive at distant sites, if they arrive in the brain, they don't get any advantage from CXCL12. If they arrive in the bone marrow, where CXCL12 is abundantly expressed, that begins to feel like home. So they were pre-selected, primed to succeed in bone. But most of the genes that we have identified that confer strong organ-specific metastatic ability are the result of selection for these traits among the populations that spend time in that organ, in that host site, and are trying to graduate to move beyond the micrometastatic state. If they are in the bone and they have the ability to activate osteoclasts, that will be selected, and so on. So, however, it's both. Priming in the primary tumor for some traits, as well as selection by the host organ.
Okay, and because we are almost done with time, but I'm going to put in one more question here because it comes from the NCI. Do we know of any differential characteristics of primary tumors that can predict development of metastasis?
There are certainly lots of signatures, transcriptomic signatures, including some of the ones that we published, that have a very strong predictive ability. That was more clearly seen in the case of lung adenocarcinoma metastases. However, there is not enough to say, if a tumor expresses this gene, this is a metastasis gene. And I think that we are not going to see that very much, because this gene-centric quest for metastases, it's fine, but it's only complementary to the cell phenotype-centric quest. It's about the cells. The tools that they use are important, but it's the cell phenotypes that are endowed with the ability to regenerate the wrong tissue in the wrong place, regenerative stem cells, turn malignant. That is the main driver of metastasis. And the content of that in a primary tumor, one might study it one day and be able to predict based on that what is the likelihood that that tumor will have seeded successfully at the time that we diagnosed it already, has already successfully seeded with a latency period or not, multiple organs.
Okay. Well, thank you, Joan, again for this wonderful seminar. You can maybe answer some of the questions afterwards. So to everybody out there, that concludes the seminar and the seminar series. I want to thank everybody who has joined us throughout this difficult time by tuning in, engaging with our speakers, and as is the MSKCC way, always providing thoughtful scientific discussions. So with that, I say goodbye, stay well and safe. And now I will once more hand over the virtual microphone to Joan for his own final remarks. Yeah.
I just want to express my gratitude and the gratitude of the entire community for those who have made this possible, the whole communications team, from video to advertising and marketing the communications, the social team, etc. Very especially to you, Andrea, and your other team members in the team of hosts, Tuomas, Omar, Danwei, and Keivan, and last but not least, certainly, to the one who made all of this possible, Ushma Neil. She has been the magic behind the scenes that has kept the whole series together, selecting fantastic speakers, except for the last one, of course. To all of you and all of them, great gratitude from all of us. Thank you all. Have a safe rest of the summer. Hope you get a break to enjoy yourselves and your families and your friends. And I'll see you around next time. Goodbye.
Goodbye.